Thank you for the title of my talk is adding a new layer of sweetness to your single-cell sequencing data. And today I'm going to talk about, of course, sweet stuff, glycosylation, glycans, sugars, and its applications, single-cell sequencing, and how we can combine these two in the same experiment. So without further ado, let's begin with the introduction of our company. So we are Singleron Biotechnologies, and we established in 2018. Since then, we have offices all around the world. What we do is we produce our own single-cell multi-omics solutions, single-cell sequencing kits. We are also providing them as a service. So let's start with what is glycosylation. It's one of the most frequent post-translational modification where sugar moieties are attached to the backbones of proteins or lipids. There are at least 5 types of different glycosylation, and I'll show them here, namely N-linked. N-linked glycosylation, these monosaccharides are binding the amino group of asparagine on the protein. With the O-link, monosaccharides are binding the hydroxyl group of serine or threonine. There are other forms of glycosylation, but the most frequently seen are the N-linked and O-linked. There is also glycation. In this case, these monosaccharides are linking phospholipids to the proteins. With the C-linked glycosylation, the molecule that binds is the mannose. It is binding to the indole ring of tryptophan. And with phosphoglycosylation, glycans are binding the serine via phosphodiester bound. This is a different approach than O-linked because there is a different bound. On the right side, let me just turn on the pointer. So on the right side, there is a nice review that you can see differences between different glycosylation. And you can see that there is a different core and different branching among different glycosylations. So this glycosylation event is taking place in the ER and in the Golgi. So it's the combined action of both organelles. So what is the importance of glycosylation? So imagine that in our system, more than 50% of proteins are glycosylated, are known to glycosylate. And there are even some reviews that says up to 70% of proteome is glycosylated. So that adds, this post-translational modification adds extra complexity to the process. And you can imagine that this can be applied or seen in various fields. One important field would be the T-cell biology. So glycosylation and the genes that are responsible for glycosylation is very important for T-cell activity and function. So it is in the center of homeostasis, proliferation, signaling, and development. So the pathways that are involving these processes are heavily glycosylated. And if there's something wrong with this glycosylation process, you can expect that it will show up as a form of disease or as a form of normality in the cell. And T cells, thanks to this different glycosylation, they have different immunogenicity and different tolerogenicity in the cancer and immune diseases. Another important field which uses glycans a lot is, of course, host and pathogen interactions. So in our cells, the cells are covered with a sheet of glycans. You can imagine there are a bunch of glycans that are attached to proteins. And viruses are expressing proteins known as lectins here. Lectins are proteins that have high affinity to the glycans. So they will specifically bind to those glycans. And so that it will initiate the infection process. This could happen vice versa. So virus can express spike proteins, and these spike proteins are also glycosylated. So there are glycan chains on the surface of this protein, and by doing that, they will basically bind to the lectins that are expressed or presented on the host cell so that they will initiate the infection. Therefore, it is important to understand the changes in this glycosylation, change in the glycosylation patterns to understand, to elaborate better these biological complex issues or questions. There are many studies on aberrant glycosylation, And basically, when there's something wrong with this process that affects more than 50% of the protein, of course, it is expected to see some diseases. And I just showed 3 of them here. And the first review article is illustrating the importance of glycosylation in liver diseases. The second one is focusing on the Alzheimer's disease. Currently, there are some glycosylation changes in the Alzheimer. And for the cancer, it is considered one of the hallmarks of the cancer. And in this review, we can see that they basically, all the proteins that are involving in different steps are glycosylated. And when there's something wrong with them, that could lead to cancer progression or tumor progression. There are different genes in the glycosylation process that affect these processes. So that was the beginning of glycosylation. Now I'll continue with the single cell and why we need single cell. So this is an illustration. I like this very much. This is from George Seurat. He's an impressionist artist from 19th century, and this is one of his paintings. So here, what you can see, that we can imagine that this painting is a tissue, and each individual pixel here is cell. So these pictures consisting of cells, individual pixels. But what you can see, if you think that this is a tissue, the information that you can get from that would be, let's say there are some blue cells. These are overrepresented cells. You can see some green and some yellow. Let me turn on my pointer so you can also see. So you can get information from these parts, and maybe some gray and kind of dark colored sky. But the thing is, you will get information from mostly overrepresented cells. With single cell sequencing, then you can see that each individual pixel, and you can get information from those. So each represents a cell. And then you will realize what you see previously in this blue area actually consists of many different colors. So this is something that you would miss if you just go through bulk RNA sequencing. So with bulk RNA, you're pulling the results and you're averaging the sample. With individual cells, you can precisely profile each cell type. That would facilitate the discovery of drug targets and biomarkers and can be used for many different purposes. So application fields of single-cell sequencing are cancer, neuroscience, immunology, and developmental biology, but you can also add much more to that. One of the main reasons why people are using, as I said, single-cell sequencing is to understand the tissue heterogeneity. Tumor microenvironment consisting of many different cells. But bulk RNA sequencing, we are basically not getting enough information from the tumor microenvironment. With single cell, we can actually go deeper and obtain deeper information from these tumor microenvironments. For neuroscience, there are many different cell types in our brain, and we can classify those, classification of neurons, and we can get information about cellular interactions. Also, we can get insights into treatment of neurological disorders by using single-cell sequencing. Another important usage of single-cell would be accurately identifying immune cell types and dissecting the gene expression patterns or cellular interactions during developmental processes. So if you look at the PubMed, if you search single-cell sequencing, then this is the graph that you will see. Number of papers that use single-cell over time is rapidly increasing. And here you can see there is a dramatic increase after 2017, and this is because the high throughput systems are introduced at this time. And after that, it really accelerates the research of single-cell sequencing. So there are several methods where you can use to detect glycans. So here I just illustrated from simple and low cost to complex and high cost. So the most basic approach to detect glycosylation would be running an STS page. So these glycans or these sugar moieties have certain mass, and when they're glycosylated, when proteins are glycosylated, they will increase their mass, and so that when you run it on the gel, you will see different bands. So this is one indication that you can see and detect glycans. There are colorimetric assays, which basically change colors in the presence or absence of the glycans. There are lectin assays, and flow cytometry is also heavily used. So just to repeat, lectins are proteins that are high affinity to glycans. So they are very targeted and very specific to certain glycans. And people are exploiting this property. For example, they are having a lectin and they are attaching biotin, or they are attaching a fluorophore to detect those glycans. The gold standard of detecting glycans is considered mass spectrometry. This is a non-targeted approach. With mass spectrometry, you can look at the entire glycome, but this is it's not something that you can do on the single cell resolution. So currently, single cell technologies or single cell have been used to tackle this problem. But the thing is, the genes that are responsible for glycosylation, they are not highly expressed most of the time. So single cell alone cannot provide the comprehensive information. And this is why we thought coming up this product that enables quantification of cell surface glycosylation together with the whole transcript of a single cell resolution. And I will just tell you more in the upcoming slides. So this is the workflow of ProMoSCOPE. It consists of 4 main steps. So first step is the cell labeling, then the cell partitioning, mRNA capture, reverse transcriptase, transcription and library preparation, and of course sequencing and data analysis. So I will go deeper in each step. So with the cell labeling, what we have is a special tag. We call it ProMoSCOPE tag. This tag contains a glucose residue in front that is chemically linked to a PCR handle and barcode that is followed by poly A tails. So we use this click chemistry approach to produce this tag. And the click chemistry is probably you already know that you got a Nobel Prize for 2022 in chemistry. It's a very nice click for biological purposes because it does not affect it doesn't have secondary effects compared to other 3D bindings. So this tag will be conjugated at the glycans at the cell surface. For that, this fructose residue and PMT conjugator is recognizing an lactosamine sequence. It's a glycan. That's a very special disaccharide. That's the universal disaccharide. So the cell surface entirely will contain this. And we basically linked this ProMoSCOPE tag to the cell surface. And after that becomes cell partitioning. So the core technology of Singleron is consisting of these microchip that you can see here. I don't know if you can see clearly on my camera. So this is the size of microchip. And here, what you see is this transparent region, this whitish transparent region that contains hundreds of thousands of microvalves. That is illustrated here. And what you do is you basically take your sample, single cell suspension, and you load it from one hole and remove from the other. And you just do some washing steps in between. So what happens is that this is a microfluidic chip. So once they're in, they will follow a certain pattern, and they will fill these holes, these microvalves. And because of the gravity, they will sink to the bottom. And this is very important because unlike the other systems, this is controlled by the gravity. So it is very gentle for the cells. Especially, this is important, especially if you're working with delicate cells, such as neutrophils. So they are gentle. So once they are separated, we load our barcoding beads. And on the right side, you can see our barcoding bead structure. This consists of several oligos, several types of oligos, namely read sequence for PCR, barcode sequence, UMIs, and poly-T. So barcodes, each bead will have its own unique barcode sequence, so that once we pull them together, we can trace it back. So we can identify the origin of the cell by using this barcode. UMI stands for, or UMI or UMIs stands for unique molecular identifiers. This is basically each transcript will get one UMI. So when we do PCR in the later step, we eliminate this PCR amplification bias by using these UMIs or by counting these UMIs because they will also be amplified. Finally, we have poly-T. This is to capture polyadenylated mRNA. But this is our own technology, so we can also modify these sequences as we want, which I will briefly mention at the end. So far what we have is this label. We label our cells, and then we load this microchip. So they will flow, they will sink to the bottom, and then we add our bead structure or with our beads, barcoding beads. So this is how they look under the microscope. So as I said, this is the size of a microscope slide. So probably every lab has a simple microscope. You can just go and see it under the microscope. This is how they look like. If you look at the empty wells, this is how they look like. And once you load the cells, here you can see that cells are sparsely distributed. And you can see that one well will get one cell. And then we'll basically cover the entire wells with the beads. In this case, it doesn't matter if the well is empty, because we only care about the wells with the cells and the beads. There are three different configurations that microchip comes with. The first one is SD, standard density. We can go up to 10,000 cells. The second one is high density chip. We can detect up to 30,000 cells. And the third configuration of this microchip is large well. And since this is our own technology, we can actually modify or change the size of these wells. So most of the cells are below 30 microns, but some of them are bigger than that. And they can be challenging to handle because they just don't fit in those either droplets or they don't fit in these wells. So what we did is basically we increased the size of this well to accommodate irregular shaped cells, such as neurons or cardiomyocytes. But this is also available. So the next step is mRNA capture. So that part, the cell lysis mRNA capture takes place in this microchip. And once we load the barcoding beads, remember that our ProMoSCOPE tag also contains these poly-A tail. So that they also will, so this tag will also be captured together with the whole mRNA. And then what we do is to separate this and prepare two different libraries. And one would be the ProMoSCOPE library, where you can get this glycosylation or glycan counting, quantification of those glycans. And the other one is the whole transcriptome from the same cell at the same time. And the next is the sequencing and data analysis part. Here, you can use any Illumina-based sequencers to sequence. Our libraries are compatible with Illumina, and a data analysis pipeline is also freely available on GitHub. So once you perform your experiment, you can analyze your own data by using these tools. So that will be the principle of ProMoSCOPE. So one of the first questions that we ask is that since this is some sort of labeling involved, we ask whether or not this affects the transcriptome, because this is always a question. If there is some sort of interaction between two proteins or two residues, there is a high chance that that will affect the conformation of the proteins. And this is exactly what happens if you use this lectin-based assays, because the lectins are also proteins and the glycans are also proteins. So basically, once they are linked to each other, they change conformation. And that often have a side effect on the transcriptome. But in our case, our tag contains a fucose residue. So it's a very similar residue. And this should not affect the glycosylation. And this should not affect the whole transcriptome. Here, what we show exactly that. So for those who are not familiar with the single cell, this is a UMAP plot. Basically, each dot here represents one single cell. And cells here are projected based on their similarity to each other. So that means similar cells will cluster together. So if you look at the first UMAP plot, you can see the cell identities. So these are the cells from bone marrow, and you can see that cells that we identified, like monocytes, macrophages, and neutrophils, all of them are here. The second UMAP plot is basically colored by either labeled or the control cells. Controls are the unlabeled cells. And here you can see that they are perfectly overlapping. This suggests that transcriptome after this labeling is not affected. And also you can look at the cell type composition in the tissue, and you can see that there is not much change between control and labeled samples. So overall, we can clearly say that with this technology, with this labeling, the whole transcriptome is not affected, that there is no side effect. So this is another analysis. Here you can see the Pearson correlation. It's between labeled and control samples. It's 99%. So it's very, very high correlation. That means there is no really difference between these two groups. So going back to that same data set, what you get from ProMoSCOPE experiment is the tag UMI counts. So here you can see, and what you realize is it is colored by the amount of tag UMIs. So the red represents around 10,000 and blue represents between 0 to 2,500. And you can see and appreciate the difference here in this group of cells and this group of cells. So let's look at what they are. So these group of cells are GMPs and neutrophils, and of course part of the monocyte. So that means these cells naturally have more glycosylation event going on in their cell surface compared to the other part of the tissue or the cells. And on the violent plot on the right side, this is also reflected. The monocytes and neutrophils have the highest TEGRMI counts. The second organ that we'll look at was the thymus, because thymus is important for T cell maturation. And so on this case, on the first plot, you see the cell type annotation. There are different cell types that we can detect. And in the middle, that's the TEGRMI counts. Here you can see also, like, for example, gamma delta T cells, and part of these groups have more glycosylation than the other type of cells. And this is also reflected in the violin plot on the right side. So this is important to know because you can see tissues are heterogeneous. And by using this bulk sequencing or bulk methods that I described earlier, there is no way that you can get this information. And this is why it's important to go single cell, but not just single cell, combine this with this glycosylation information. So in our tech, so the technology that I described you belong to according to research needs. So by using that, there are different technologies that we can offer. The second one is the human VDJ profiling, immune VDJ profiling. If you're a immunologist and if you're interested in knowing the clonal expansion of T cells, then this is the right tool for you. And there are another unique tool that we come up with. We call it ProMoSCOPE. What we do is we design specific sequences that captures specific genes. But when you capture those genes, you can specifically look at the genetic variants, fusion genes, and viral targets. So viral targets are also important because often case, they are not highly expressed. And with this technology, you can capture the transcripts from the very early stage of right after mRNA. So there will be a specific enrichment in the beginning, and later on, there are also some enrichments. So you will at the end get very high sensitivity by using this technology. So with this one slide, I want to explain what ProMoSCOPE is about. ProMoSCOPE can detect gene fusions, viral sequences, and single nucleotide variants together with the whole transcriptome. So this is very important. That's why it is very different from other targeted panels, because with the other targeted panels, you're not getting any genetic information or gene fusions, etc. With ProMoSCOPE, we do this thanks to our unique barcoding beads. We do this with high accuracy and sensitivity. If you're dealing with the rare transcripts or lowly expressed genes, we can design specific probes for those to enrich them. We are also not limited to polyadenylated transcripts. So this bottleneck of single-cell sequencing is basically most of the capture takes place by the poly T and poly A. But with ProMoSCOPE technology, we are not limited to this. We can also detect other type of RNAs, even though they are not polyadenylated. So with these technologies that I described you can think about like a layering up your single cell data. So first and the most basic that you will get is the whole transcript of information. For most of people, it's enough. But there are some people, they want to do more. They want to explain their data better. And then you can basically go one layer up and you can detect the mutations if you're interested in certain mutations. Because for cancer, especially for certain cancers, there are druggable mutations that significantly change the outcome of the diseases. Therefore, it is very important to understand what's going on in those cells with the mutation. So this is something that we can do. And you can go one level up, and then you can also look at cell surface glycosylation together with the whole transcriptome and mutation information. So basically think about this as a layering up your experiment. Or if you're, let's say, if you're an immunologist and you want to know the clone information of T cells, then you can combine this information with the glycosylation, then you will have the bigger picture, basically. So whatever I describe you today, all the technologies, this is our own technology. We produce this and we provide kits for you to perform this experiment in your lab without needing any instruments. This is very important. As I said, you don't need any instruments to perform single cell experiment. All you need is just this microchip, which you can use for your research. But we are also offering all of these services as all-in-one worry-free service. We have a service lab here in Cologne, Germany, and basically you send us your sample, we take care of all the downstream experiments, analyses, and at the end you will get your publication-ready results. So this is a really worry-free approach if you want to try our services. And the reason why you should work with us is we provide end-to-end solution. One of the most important steps, if not the most important, is basically obtaining viable single cells from the tissues. And from this perspective, we have more experience with more than 400 different tissues. So we can get viable single cells from those challenging tissues, even the liquid biopsies, which is very difficult to obtain and difficult to process. But we have experience with those as well, with a very high success rate. Of course, you will get a PhD level support and you will get your own dedicated project manager to your project that will follow the entire project with you and keep you informed during the process. So there will be always a connection. And we offer this service in the entire European Union. And we have really fast turnover time. It's around 6 weeks, including data analysis part. So these are the reasons why you should work with us. And with that, I would like to conclude my presentation and thank you for your attention.
Thank you again, everyone, for your time today. And if you contact us by 15th of December, due to our product launch, you can get a special price from us. But you can visit our website and also follow us on LinkedIn and Twitter by using these QR codes that you can see on the screen. Have a great day.